Today we will discuss about mitochondria. Mitochondria are granular or filamentous organism present in eukaryotic cells. In this diagram you can locate them as labeled as number 9. These are the organelles. They will be present in multiple number within a cell. These organelles were first observed by Fleming and Kollam in 1882, and the term mitochondria was used first by Benda. These organelles are responsible for energy metabolism and carry the enzymes responsible for this metabolism. As they deal with production and storage of energy, they are called the powerhouse of cells. This is achieved with the help of ATPase and ATP synthase enzyme. Number of mitochondria present in a cell depends on the physiological status of the cell. In a metabolically active cell, number of mitochondria will be more as it needs more amount of energy, but when the cell is older and metabolically inert, number of mitochondria will be limited. These mitochondria are distributed throughout the cytoplasm in normal cell. Sometimes they are aggregated towards the nucleus, and they are also seen towards spindle apparatus during cell division. Mitochondria can do protein synthesis within it. For that, ribosomes are present inside mitochondria, and such ribosomes are called mitoribosomes. And this is the schematic view of mitochondria. This is a structure what we are going to explain. They are having a elongated structure, and this is the outer membrane, and this is the inner membrane, which has formed into so many infoldings. These infoldings are crystal. We'll go one by one. So mitochondria are filamentous or granular structure. Though we tell like they are rod shaped, many times they are not only rod shaped, but they can be ring shaped, rounded or vesicular. They can be club shaped. So mitochondria occur in different shape on different organisms. Similarly, size of the mitochondria is also variable. Generally, the mitochondria has a length of 3 to 10 microns. These mitochondria are having two membranes, an outer membrane and an inner membrane. The outer membrane is called outer mitochondrial membrane, and inner membrane is called inner mitochondrial membrane. These two membranes are lipoproteinaceous and unit membranes. Each membrane is having a thickness of 60 angstrom, and these two membranes are separated by a space of 80 to 100 angstrom. The space between the outer membrane and inner membrane is called perimitochondrial space, and it is filled with a fluid which is having less viscosity and very low density, and it is called a perimitochondrial fluid. Coming to the membranes, outer mitochondrial membrane is freely permeable, while the inner mitochondrial membrane is selectively permeable, or it is impermeable to most of the molecules or ions. Around 70 proteins or enzymes are present within the mitochondria. They include coenzymes also. They are located within the matrix of the mitochondria or within the membrane. These enzymes are mainly used in cell respiration. Mainly the enzymes in our inner membrane, or the enzymes located on the inner membrane, are respiratory enzyme. And as an example, we can tell ATP synthase is such an enzyme. for the production of ATP. So this is the typical structure of a mitochondria. This is the outer membrane and this is the inner membrane. This is the outer membrane and this is the inner membrane, and this space between these two membranes are called the perimitochondrial space, and this space is filled with a fluid, and this is the inner cavity of the mitochondria, and this region is called the matrix. The inner membrane of the mitochondria carry carrier proteins. They have carrier proteins, and such carrier proteins are called translocases. They are involved in the transport of glutamate, phosphate, ATP, ADP, etc. At some region, the outer mitochondrial membrane and inner mitochondrial membrane will come in close contact with each other. Such contact points are called contact zones. And these contact zones are the regions of transport of proteins from cytoplasm to mitochondria. Many a times, proteins need to be transferred from cytoplasm to mitochondria, and it is carried out through these contact zones. The inner membrane enzymes are concerned with redox reactions and oxidative phosphorylation, whereas the enzymes in the outer membranes are not concerned with redox reactions or oxidative phosphorylation. As I told you, the space between inner and outer mitochondrial membrane is called outer chamber or perimitochondrial space, whereas there is an internal cavity within the inner membrane. Within the inner membrane, you can see an internal cavity. This internal cavity is called a matrix of mitochondria, and it is filled with a fluid, and that is called a mitochondrial fluid or mitochondrioplasm. These matrix contain dense granular structures. If you see this inner mitochondrial membrane, this is the inner mitochondrial membrane, and it forms invagination or foldings into the inner chamber of the mitochondria. So many foldings you can observe. These foldings are called cristae, and these cristae are the structures which carry the enzymes for redox reactions and oxidative phosphorylations. The mitochondrial membrane contain stalked particles, and these are called F0 particle or elementary particle or ETS or oxysomes. In the inner membrane, they are found to be stalked. There are reports like this. Oxysomes or elementary particles are present in the outer membrane also, but they are not stalked, but which are present in the inner membrane are stalked. If you see the structure of this oxysomes or inner mitochondrial oxysomes, or the tennis racket or stalked structures within the cristae or inner mitochondrial membrane, they are having a base piece, a stalk piece, and a head piece. See the diagram. They are having a base piece, they are having a connecting stalk piece, and a head piece. These three pieces are having a distance of 100 angstrom apart. So they are having a distance of, from base piece to stalk they are having 100 angstrom, then stalk piece to head piece they are having 100 angstrom. The base portion is hydrophobic and it is embedded in the inner mitochondrial membrane. It is formed of polypeptides and proteolipids. And the stalk piece, it connects the base piece with the head piece, and they are called as F5 or F6 particle, and it contains oligomycin sensitivity conferring proteins. They are concerned with binding the particle with mitochondrial membrane. The head piece is the most significant one. This is hydrophilic and capped with an ATPase inhibitor. So this is the ATPase inhibitor. When there is ATPase inhibitor, this enzyme will not work. Otherwise, if the ATPase inhibitor is removed, it will work. The headpiece is formed of 9 polypeptides. It's a protein. Of course, it is ATP synthase and it is formed of 9 polypeptides, 3 alpha chain, 3 beta chain, and 1 delta chain, 1 epsilon chain, and 1 gamma chain. This is the structure of a typical ATP synthase called as F0 particle or oxysomes. The shape of the cristae also vary. Normally we are telling an infolding like this, but the shape of the cristae can be reticulate depending on the species. Chemically, mitochondria contain 65 to 75% proteins. They have 25 to 30% lipids, 0.5% RNA, and small amount of DNA. The lipids present within the mitochondria have 90% phospholipid, 5% cholesterol, and 5% fatty acid. Apart from these molecules, they also contain sulfur, iron, copper, and some vitamins within the mitochondria. The mitochondria are called semi-autonomous organelles since they are having DNA, and the mitochondrial DNA present within the mitochondrial chamber is circular, double-stranded, and self-replicating. And ribosomes also present within the mitochondria, which will help them to do the protein synthesis. Different hypotheses are there regarding the origin of mitochondria. One such hypothesis is during maturity a mitochondria will enlarge and finally break into pieces, and each piece will form a new mitochondria. Second one is from endoplasmic reticulum or plasma membrane, the outgrowth will be formed, then there will be influx, and as a result it will be converted into an organ, and that is the mitochondria. This is called the origin from endoplasmic reticulum or plasma membrane. The third one is called de novo origin. It simply originates. How it happens, it is not described. The last one is prokaryotic origin by Altman and Schimber. According to that statement, it is a mitochondria is a prokaryotic structure. It migrated onto the cell and lives symbiotically with the host cell. These are the 4 theories regarding origin of mitochondria. Coming to the functions of mitochondria, the mitochondria include thermogenesis, where they help to maintain the temperature of the organism. They do protein synthesis since they are having ribosome. They do the synthesis of steroid hormones. They are having mitochondrial DNA, RNA, and proteins. They are involved in urea cycle. The first step during urea cycle where ornithine is converted to citrulline, this happens within the mitochondria. They store calcium, so it helps in calcium accumulation, mainly cation accumulation. They are involved in energy supply, and cell respiration happens within the mitochondria. These are the major functions of mitochondria. So, to conclude, mitochondria are the powerhouse of the cells. They are having a membranous structure, 2-layered membrane, outer membrane, then there is inner membrane. Inner membrane is having infoldings. The space between 2 membranes are called a perimitochondrial space, filled with a fluid called a perimitochondrial fluid. Inner to mitochondria there is a space called a chamber. It is also filled with a matrix. Now, coming to the inner membrane, they are having lot of tennis racket like bounded structures called oxysomes, and they are made up of multiple units: head, stalk, and base. And the head is mainly ATPase, and it is formed of 9 particles. These organelles do the cell respiration, energy supply, and related things within a cell. Thanks for your patient listening.